I'll invite Professor Arvind Rao to speak on spatial bioinformatics tool for tumor microenvironment. Mr. Arvind Rao.
Thank you. Thank you. Hello, everyone. I hope I'm audible. So my name is Arvind. I'm faculty in University of Michigan, Ann Arbor. Firstly, thank you to the organizers for inviting me to present today. I'm going to be taking a bit of a shift from the previous conversations, talking about computer science, statistics, and bioinformatics. As you might acknowledge, bioinformatics is a requirement nowadays, given the vast amount of biological data coming through. Arguments have been made that biology is the new physics, a lot more data, lots of opportunities for quantification. So I'm going to start with that as the premise. I'm hoping the students in this audience will enjoy this talk, and if there's any questions, I'm happy to talk after this presentation as well. Once the slide starts working.
It's not moving.
Yeah, I realize that. Anyway, I can start talking about it. Hopefully, it'll make sense as we go through things. So the promise of this entire conversation starts with immunotherapy.
Speak from the podium.
Oh, it starts with immunotherapy. Yeah. So to this group, it's not a surprise. Immunotherapy is a very important modality for cancer care, and so there have been a lot of interesting aspects of how immunotherapy can be used as a fourth prong, and it's got the Nobel Prize a few years ago. The aspects of microenvironment manipulation matter both in oncology and in developmental biology. There's a lot of similarities in stem cell biology that could be sort of used for learning across disciplines. I have to say it's a very hard act to follow after Dr. Kaul and Dr. Vishwanatha, but I'm going to try. It's a bit of a detour, but hopefully what I'm going to say is going to be in line with what we just learned from their talks. So the whole idea is that you have this extensive heterogeneity, which is a hallmark for the variety of disease processes in the microenvironment. There's intratumoral heterogeneity, which is inside each tumor is unique, and between tumors, between patients, there's a lot of uniqueness as well. And an argument can be made that these tumors are actually ecosystems. I want to just dwell on this aspect a little bit. Think of the continent of Africa, or think of your favorite continent, it could be India, right? You have an exact same, it's an amazing, incredible complexity. Like you have Rajasthan, you have a desert, you have, right next to it, you have a few states that are extremely green, you have lakes, you have tributaries, you have rivers, you have oceans, all skirting this beautiful continent, right? What does it tell you? It tells you, and different, depending on where you are in the country, you have habitats, extensive forests, extensive sort of habitats for wildlife of different kinds, some bird sanctuaries, some tiger sanctuaries, all in the same continent, right? What's happening? It's a very beautiful interplay of weather systems, water systems, and ecology. That's the beauty of ecology. An argument where ecology can go bad is in tumors. What's happening in tumors? There are ecosystems of different cell types that like to eat other cells, cell types that like to get eaten by other cells, immune cells, fibroblasts. All these things are an extensive ecosystem. So the question is, nothing is operating in isolation, right? These are all reciprocal interactions between different cells, and can we find ways to quantify them using new technologies? That's the whole premise of my talk. So data like this is becoming extremely available. This is data from Gary Nolan's group in Stanford, where he's used a very interesting technology called Codex, which is like doing proteomics with about 40 different markers on a piece of tissue. So what you're seeing on the right-hand side is a piece of colorectal tumor, right? And all the colors you see are different cell types. That is the incredible complexity of that tissue, right? And we want to be able to identify what can we learn about these things spatially, and can those spatial measurements give us any clues of how to treat the tumor? Because as we just said, tumors are unique, right? So I used to like playing with Legos. I'm sure some of you do. So Legos are the following, right? You have these building blocks. Thank you. you have these building blocks. Is this any better? I'm not able to find the bottom. Okay, how about this? No? Hello? Okay, thank you. So I'm a Legos guy. Look at these Lego blocks, right? If you had data, like cells coming from these Legos, right? You can sort them, you can arrange them, you can do all kinds of nice things with them. But if you didn't know they came from this nice little house, like a spatial context, you would never know what these blocks mean. So spatial architecture is critical, right? For understanding things like microenvironment. Tools like this are becoming online and is incredible amount of data, but no one's got a very good clue on how to analyze this data, right? You can run from this kind of data, but you can't hide, because at some point this data is going to come right into your face and you have to figure out how to analyze it. So if you want to make sense of genomics data and spatial transcriptomics, this is probably a great opportunity for young scientists. So we are using statistical machine learning tools. All of you have read about and learned about ChatGPT, you've seen AI, it's very big now, right? So what can we do with these tools in this context? The humble unit of measurement for this kind of quantification is this kind of spatial multiplexed image, right? Where what you see is a piece of tissue, a single piece of tissue, actually lung cancer, and every single dot you see is a different kind of cell type, okay? So what you're seeing is now an incredible complexity in the spatial configuration of different cells. This is what you see in an image. How do you quantify this? Can you make any measurements? So we built a toolbox that lets you do a few of these things. You take a piece of tissue and you give me imaging on those. Yes? Talk even louder? Okay. Talk even louder. All right, I'll try. Okay. This will work? Okay. All right, let me try this. So we built a toolbox that lets you do spatial proximity identification and then connect that to things like outcomes. Please give me a 2-minute buzzer. That way, if I run short, I'll speed things up. But the idea comes from the following idea. I'm going to try to give you a cartoon, right? If you get this cartoon, basically you get the premise of my talk. You got two different situations here. The circles represent two different pieces of tissue. The left is what is called a cold tumor. The right is what is called a warm tumor. Why is it warm and why is it cold? Because on the left-hand side, the tumor cells in red do not talk to the immune cells in blue. They don't want even, they have no interest in each other. They don't engage each other. On the right-hand side, which is a warmer tumor, there's a lot more engagement between these cells. So if your favorite technology is anything that is not spatial, like single-cell genomics, bulk sequencing, good luck trying to differentiate these two situations on your favorite sequencing platform. You have to go spatial, right? That's obvious. By construction, the number of cells that I have for immune cells and tumor cells is exactly the same. So with single cell genomics, you could not tell them apart. You could if you had high coverage, but in most cases, clinically, it's not possible. So you have to do something spatial. So if you did, like I said, gene expression, it gives you the same answers. And if you went to a pathologist in France, and the reason I say France is because this paper came out from Jérôme Guéllot's group in France, where they said, we can do this quantification spatially, but we need a few things. We need highly trained pathologists whose only job is going to be finding areas to put square windows, and in those square windows, we will count the number of immune cells. They call this the immunoscore. It's a very popular idea. It's a very great idea, provided you have pathologists available in every nook and corner of wherever you want to practice. So it's not scalable. If it's not scalable and you want to make it objective, I would argue that computer science and AI is probably a good way to be able to scale that up, right? The good thing about AI, my advisor used to always tell me this, is AI is at least reproducibly wrong, right? There's no variability in AI in the sense that once you have an answer, even if it's wrong, at least it's consistent about it. So you can use that to your advantage. So what we've done is with things like spatial statistics, I'm going to cut, if you want details, we can talk later. But we built a spatial statistic, which is again not my creation. It's been there in the ecology literature for almost 70 years. We simply borrowed it. And we built these curves that give us a sense for, how much mixing there is. So what you see are two red curves in the bottom, one red curve under the left, one red curve under the right. And the faster this curve rises, the more mixing there is between the red and the blue cells. That's a simple idea. So the area under the curve is a direct reflection of how much mixing there is between these cell types. More the area under the curve, the more there is mixing. Less the area, lesser the amount of spatial mixing. Why is this useful? Because now I give you a number. You wanted quantification, here you go. I give you a number that connects this image with something quantifiable and objective that you can then use to connect to clinical outcome. What did we do? We took some pancreatic cancer specimens. You see 3 different situations. This is real data, where you've got tumor cells in green and immune cells in red. And this is work with Jim Allison, who some of you might know, won a Nobel Prize a few years ago in immunology. And this is his colleagues, Raghu Kaluri, Valerie, Julian, and Pedro. And what we did was we said, well, if we can quantify this, can we connect these measurements of physical spatial proximity to clinical outcome on the right? Clinical outcome as given by survival. So I'm showing you a Kaplan-Meier plot where the green curve on the top, sorry, it's not a Kaplan, it's just the curves. The green curve on the top represents the most mixing scenario on the top. The red curve is the middle, the yellow curve is the bottom. So the more the amount of mixing, the more the curve rises. Okay? That's the idea. Very simple measurement. Once you have these numbers, you can do what any biostatistician likes to do, which is to build multivariate models that connect an image-derived number with clinical outcomes like survival. So you can build this kind of Kaplan-Meier statistic. We showed that it's independently prognostic after adjusting for clinical correlates and demographics. So it stands on its own. So spatial measurements of tumor immune configurations have something unique to offer while prognosticating patient outcome, right? Even after adjusting for things like gene expression. This is another use case where you can connect, you can look at spatial configuration and try to connect that to looking at high-grade versus low-grade disease. We've done that in pancreatic cancers. We've done this for pancreatic disease, for inflammatory breast cancer. We've done this for lung cancer, all published. None of this, what I'm telling you, is new in that sense. But what's very interesting is a very simple spatial statistic can do all this for you. I didn't even use AI just yet. I'm going to skip this in the interest of time, but I'm going to also go to this notion that with tumor heterogeneity being a very big component, understanding tumor subgeography is critical, which is, think of a tumor, there's a core tumoral component and there's a peritumoral component. Right? The core is where the center of the tumor is, the peritumoral is the stuff around it. And if you think of it biologically, they are 2 very distinct biological entities. The core tumor is a place where the tumor reprogramming has already happened. It's a stable state, right? But the peritumoral region is an area of active reprogramming of the tumor and its surrounding environment. So geographically and biologically, it's a very different entity. Hence, we basically tried to measure, and again, this is not my observation, this is Jerome Gallow's observation, and again, we simply tried to make this more objective. Right? I'm a computer scientist and a statistician, so we just try to make these things more objective. And so we're now looking at ways to be able to automatically break the tumor map, which you see in the center, that's the second from the left, into 2 separate regions, all done computationally. You don't need a pathologist in your area to be able to do this. A computer can do this, right? It breaks the tumor into a core tumor, peritumoral component, and you can look at measurements of tumor proximity. Again, mine is not the only game in town. There are multiple other people that have tools like this, but I would argue that quantification has still lagged behind. So that's the part we are trying to help with. 2D obviously asks the question, why only 2D, why not 3D? So the answer is yes, we can do this in 3D as well. And we have some work going on with collaborators that let you look at 3-dimensional stacks of tumor tissue and look at the correlation at a 3D level. So extending this to 3D is not hard. So 2 minutes, thank you. So what I've shown you in the first part of the talk is a framework that can let you look at spatial analytics for different immune tumor proximities. And our overall goal is to be able to combine tissue-based imaging with radiology for risk prognostication. With the 1 minute that I've got left to go, let me tell you something that you can't have a conversation about AI these days unless you talk about deep learning. So let me tell you what we did with deep learning. This is for anybody who's interested in the field, but if you're a computer scientist or statistician, I think this is the time to get into biology, right? Because there's so much of cool stuff with computer science that you can think about for spatial biology that we need people who can think at that intersection. So with deep learning, here's the problem, very simple idea. we have 6 different kinds of pancreatic disease, ranging from chronic pancreatitis all the way to pancreatic cancer. And we have just tagged 3 different cell types: epithelial cells, Tregs, and cytotoxic lymphocytes. And we get point configurations like this, where every color shows you a different cell type. And long story short, you can build a deep learning model, and you can get a pretty good accuracy for predicting different grades of disease. So clinical is great, it's fine. But the first question that I want to always ask my students is, well, what insight did you get about the biology that you didn't have before? And that's the interesting part here. This I find very exciting. So we looked at what the deep learning algorithm is looking at to make a decision between different grades of disease. And the answer is going to be anticlimactic to any pathologist in the room, even a biologist. It turns out that the cells that have the most weightage for discriminating disease grade are cells at the tumor-stroma interface. No surprises, but an algorithm discovered this independently. We gave it no information. That's the power of deep learning in today's times. Okay, so I want to leave you with that and close with just a notion that we do a whole bunch of spatial transcriptomics. I would love to talk to you, Dr. Kaul, about ideas around spatial biology, but I'm going to cut very quickly to the end and tell you that we do a variety of different things with spatial transcriptomics, integrating different data. If you want to talk or collaborate, I would love to hear from you. But what I want to close with is the need that data is becoming plentiful, platforms are becoming great. The problem is reliability, quantification, auditing, because garbage in, garbage out. Don't expect ChatGPT or your favorite algorithm to give you an answer. They hallucinate plenty. They come up with random answers that mean nothing. So we need a good tie between biologists, experimentalists, clinicians, and computational people to ensure that things aren't getting leaked in the process of iteration. I'll leave you with that, thanking you for your interest and the following agencies for their funding and my colleagues who've taught me all this stuff. The mistakes are mine. Whatever I've learned is from them. But thank you for your attention.